this webinar is being live streamed. I have to find you again. This webinar is being live streamed. This webinar is being live streamed, and you should have received a notification on your screen to that effect. Why we are doing this? It is because our seminars in this research group are soon going to be so popular that we simply need to live stream them. But this is also a preparation, as I am trying to figure out how to do webinars for the Microbial Ecology and Water Engineering Specialist group that I think you should all have received a notification on, and I encourage you to participate in. So this is a little chat of for the people downstairs. Please type in the chat function, yes, I can hear you, or no, I cannot hear you, or the sound quality is good or the sound quality is bad. So can somebody downstairs maybe use the keyboard and type something in the chat function? And once I get some confirmation that you can hear, sound is fine. Thank you, Adam, who is obviously in charge of downstairs. So without any further ado, let's see if we can make this now work. So it's my pleasure to introduce Dr. Tal Elad, who is a postdoctoral researcher in at TTU Environment. Tal did his undergraduate and graduate studies at the oh my God, Tal did not send me a CV.
Yeah, I forgot. Sorry.
So Tal, why don't you give me why don't you give a very short introduction of your academic credentials in a minute or less as you start. But the main thing is that Tal will provide a introductory talk on what metagenomics and metatranscriptomics are, and maybe also, you know, what they can be used for within the type of studies that we might be engaged in. So we anticipate that this presentation will be last anywhere from 20 to 30 minutes. If you have questions, please put them in the chat function. Tal will not respond to the chat questions during his talk, but we'll address them at the end of the talk. This is the way webinars usually work, and that's how we'll do it. Now, we will also, because we are actually very close to the people who are joining this webinar downstairs, we actually will come down in 40 minutes or so to have some breakfast, and you can ask us more questions. But it would be nice if you also, you know, kind of use the Q&A as we go along. So I'll turn it over to Tal. I'll turn off my camera, and I'll come back at the Q&A, where I will, by the way, look at the Q&A questions, Tal, and then transfer them to you, so you don't have to look at the Q&A.
Okay.
Okay. Mm-hmm. It's all yours.
Okay. Well, thanks, Bart, for this. To complete what you started, so I did my previous studies in the Hebrew University of Jerusalem, bachelor, master's, and PhD. And now, well, I'm a postdoc here, and I'm about to give this introductory talk to metagenomics and metatranscriptomics, kind of one-on-one. And hopefully, after this presentation, you guys will be more informed than you are now about these topics. I should say that before I start that I'm doing metagenomics. I mean, I didn't do metagenomics and metatranscriptomics before I got here. It's something that I learned to do here, and that I'm still learning how to do and how to do better. And yeah, so it's also a learning process for me still. So let's begin. So I'm going to just a simple outline of this presentation. So I'm I'm going to explain what metagenomics is, what it is good for, and then I'll give some examples from the literature. And then we'll dive into metatranscriptomics, and I'll also try to explain what metatranscriptomics is good for, and I'll give some examples from our research here in our group, and then just a short summary. So let's start with the definition of metagenomics. Well, metagenomics, according to Wikipedia, is the study of genetic material recovered directly from environmental samples. In the literature, you find similar definitions. For example, here, metagenomics is the application of modern genomic techniques to the study of communities of microbial organisms directly in their natural environments. Of course, bypassing the need for isolation and lab cultivation. Now, what these two definitions have in common is, of course, the fact that we are extracting genetic material from an environmental sample. Now, in that sense, 16S amplicon sequencing can also be considered metagenomics. Although this presentation will be about what is maybe more accurately called shotgun metagenomics, which usually we just call metagenomics. And then why shotgun metagenomics? Well, shotgun metagenomics because this is the sequencing method that we use. It is called shotgun sequencing when we do shotgun metagenomics, or simply metagenomics. And this is the principle. Well, you have your environmental sample with, of course, the microbial community. You extract the genomic DNA, and then what you do, you actually make fragments out of it. That's why I'm pretty sure it is called shotgun. And then, so you have these DNA fragments, and then you sequence them all. And the output, I mean, you started with this environmental sample with the microbial community, which is in it. And the output of this shotgun sequencing is these digital DNA fragments. They are called reads, about usually 150 base pairs plus minus long. And this is what you get. So you start with this environmental samples and you end up with these reads. Millions of reads, of course. And now the question is, okay, so what can we do with it? So what can we do with these reads? Well, two main things. The first thing is called read recruitment or mapping. Now, this is sometimes overlooked. I mean, usually when people talk about metagenomics, so they would talk about perhaps the second point here, which is the assembly of genomes or draft genomes, which is the really cool thing that you can do with metagenomics. But before that, you can simply do read recruitment. You have all these reads, and you can use reference databases like NCBI, for example, to actually map the reads to recruit them to reference existing databases, and construct taxonomical and functional profiles. And of course, the second thing, which is perhaps more talked about and more cool, is the de novo assembly, which is us taking all these reads and actually assembling genomes. So we actually get kind of the genomes of the microbes that are actually that are in our environmental sample. These are, of course, are not exactly referred as genomes, but rather as draft genomes, or more commonly, metagenome assembled genomes, which are called MUGs. So let's go through these 2 points. So, let's start with read recruitment or mapping. And here I have 2 examples from the literature. You see this paper in Nature, probably published a while ago. And you see here basically the relative abundance of the different genera in this specific environmental sample. And it was calculated by taking the reads and mapping them to an existing database, probably a very big database. And according to how many reads were mapped to each of these genera, you can actually calculate their relative abundance, which is what you see here, or also called the taxonomical profile of the community. Now, you can do the same with functions. I mean, when you recruit the reads to your reference database, you don't have to look at the identity of the microbe. You can also look at the annotation of the gene, or rather its function. And then you can draw this functional profile of the community. And here you can see, rather than the relative abundance of the different genera, but the relative abundance of the different functions. And this way, this from the same paper, you can also construct a functional profile of your microbial community. Now let's move on to de novo assembly. So as before, we do shotgun sequencing, and then we get these reads. The thing is that here you have, for illustration reasons, of course, these reads are colored based on the genome that they came from. But when we get the reads, of course, we don't know to which genome they belong. We just get, well, reads. And now we need to engage in the process of, okay, so we have this read, so let's try to reconstruct the genomes. And this is a pretty complicated and fascinating process. And you can see it here in this cartoon. You can see some basic terms. So we have these genomes in our environmental sample. We do shotgun sequencing. We get these short reads, about 150 base pair long. And then we do what is called de novo assembly. We take these reads and we actually make contigs out of them. So these would be longer sequences, about thousands of base pairs long usually. And we basically assemble these reads to these contigs. These are called contigs or scaffolds. And now, of course, remember, they are not, we don't know which contig belongs to which genome. So that's why after the shotgun sequencing and the de novo assembly, we do what is called binning. We take these contigs and then we separate them, we divide them to genomes in a process that is called binning. Now again, if you remember, these are not exactly genomes. They are draft genomes or metagenome assembled genomes, or MUGs. I also want to refer you in this opportunity. This cartoon is actually, you can see here, it's from YouTube, but it's not just one of these very simple and short YouTube kind of videos. It's actually from a seminar series, a pretty long one given by guys from the university in Chicago. I think it contains about 6 or 7 lectures, about 1 hour, 1 hour and a half long. And if you want to dive into this subject more than it's pretty recommended. Okay, so how do we do this binning? How do we know which contig belongs to which genome? Or how are contigs binned? So here we use different signals. I think that you can say that some of the more important ones of these signals that help us beam the contigs into genomes or mugs, these would be, first of all, the coverage. Now, the coverage is actually simply the number of reads that are mapped to each, that are recruited or mapped to each contig. And basically, the higher the relative abundance of the genome, the higher the coverage will be. And also, if contigs belong to the same genome, so their coverage, the number of reads that map to each to the contig pair, normalized by its length, will be the same. So contigs with the same or similar coverage probably belong to the same genome because they have, well, the same relative abundance. We also use the GC content, also a very strong signal. And another signal is what is called tetranucleotide frequencies. And I want, well, GC content is pretty much speaks for itself. I think just contigs with similar GC content will be beamed together. And let me explain in the next slide or slide and a half, 2 slides actually, the tetranucleotide frequencies concept. It's pretty nice. So what is tetranucleotide frequencies? So people found out that, well, first of all, what is tetranucleotide? Well, tetranucleotide are actually all combinations of 4 base pairs that you can think of. If you try to calculate, you will figure that there are 256 of those. It's just 4 by 4. It's like 4 times 4 times 4 times 4, right? So you have 256 different arrangements of the 4 bases. And with tetranucleotide frequency, you just take, for example, the first combination, and you look for this combination in your genome. And then you put, then you just count it, and you put this number here. And then you do the same for all of the other combinations. And it turns out that that contigs from the same genome will actually have the same tetranucleotide frequency pattern. So this is also a very nice, I think, kind of a concept to try to beam these contigs into genomes or mugs. And this is illustrated here in this picture. It's, yeah, I just couldn't resist putting it here. It's almost artistic. So you can see each dot here, it is taken from this paper, and you can see that each dot here is actually a contig or scaffold. And they are actually, oh, what's the word? Clustered. Yes, they are clustered together based on their tetranucleotide frequencies. And you can see how they actually cluster to form different genomes. Okay. Okay, so now once we have these MAGs, these metagenomic assembled genomes, so let's see what we can actually do with those. So first of all, and this is very nicely illustrated in this paper, we can construct the metabolic potential of key functional members. So once we have these MAGs, these genomes, so we can annotate. So then we can annotate them. Well, we can classify them, and then we can annotate them. So we get these kind of. So we know, for example, so in this community, so we had these 7 microbes, and we can also know what they are capable of doing based on annotating their genome. And this is very nicely illustrated here. Now, moreover, we can not only identify the metabolic capabilities of the different members of the community, we can also try to predict their interactions. And this is again a figure from the same paper showing predicted interactions between the different bacteria, or rather the different MAGs that they detected in their environmental sample. So you can see here, for example, these bacteria, they take up these carbon sources and will produce these electron donors for these bacteria. These, well, in this case, anaerobic bacteria. These would be like kind of, nitrate reducers and sulfate reducers and so forth. Okay, so what we saw here, that we can use these MAGs both to identify the different microbes in our environmental sample, their metabolic capabilities, and their interactions. And this is by classifying the MAGs and then annotating them. Now let's move on to metatranscriptomics. So what is metatranscriptomics? Now, to put it simply, if we take the metagenomics definition and we just replace DNA with mRNA, so we kind of get what metatranscriptomic is, which is the study of mRNA recovered directly from environmental samples. Now, the process is in principle the same. And then, of course, the big difference is that instead of short reads of DNA, we get short reads of RNA. And of course, we do this in order to study the gene expression. Now, of course, all this may sound simple, but of course, it's not a very simple process, especially, and if I'm not mistaken, for example, extracting DNA, let's say it's fine, but extracting RNA, it's a bit more complicated, but let's assume it's simple, and then take it from, okay, so now we have this RNA short reads, and okay, let's see what we can do with those. So if metagenomics kind of answer the questions of who is there and what they can do, and also as importantly, how they interact potentially. So with metatranscriptomics, we can actually get information about which genes are expressed and to what extent. And now let's dive into these examples from the research done in our group to maybe explain this concept of metatranscriptomics better, or rather what we can do with the RNA reads as opposed to the DNA reads. So what we have done, or more specifically CK, which was I'm pretty sure visiting PhD here in our lab. So she operated this Anamox reactor, this SBR. It was an intermediately aerated, as described here in this simple figure. And it was loaded with nitrogen. It was a pretty high load of nitrogen. It removed it very efficiently. And then one of the things that CK did was to take samples for DNA and RNA extractions. And she took them up during the aerobic mixing and during the anoxic mixing. That would be when the reactor was aerated and when the reactor was not aerated. Again, this reactor was intermittently aerated in order to maintain Anamox activity. So there were periods where the reactor was not aerated and periods where the reactor was, periods when it was aerated and periods when it was not aerated. So referred here as the aerobic mixing and the anoxic mixing. And then, as we discussed, so CK and also Alex and Marlene, they extracted the DNA and the RNA from these samples. They did shotgun sequencing, and then we got these reads, these DNA and RNA reads, millions of reads, and for analysis. And this is what I'm going to get into now. Yes. So first of all, we wanted to construct the kind of to find out, okay, who is there and what is their relative abundance. And this is here in orange. So basically, we took the DNA reads. We did the de novo assembly process. We assembled contigs, and then we assembled mags. And here you see the most abundant mags based on the calculation of the relative abundance. Again, I remind you, this relative abundance is calculated based on the coverage of the mags. That is, how many reads each mag actually has per its length. So the more the reads per base pair, the more the relative abundance, of course. And then we actually extracted 68 different mags from this environmental sample from these reactors. Here you can see the most abundant ones, and you can see that we extracted 4 AOB mags. These were classified as AOB, so you can see AOB1, AOB2, AOB4 here, and where's AOB3 and AOB3 here, and 2 Anamox mags. Here you can see them. I mean, these are mags classified as Anamox, and you can see them here, Anamox 1 and Anamox 2, where the rest are probably heterotrophs. And you can see the Anamox mags and the AOB mags right up here among the most relative microbes in the culture, as expected. Now, the other thing that you can see here is actually what we call here the relative activity. That would be similar to the relative abundance, only calculated using the RNA reads. So this here we actually map the RNA reads to the mags and investigate their coverage based on the RNA reads. And here you can see the picture is a bit different. In terms of number of RNA reads per base pair of the genome, of the genome length, you can see that the Anamox mags, the Anamox microbes were very active. Yeah. So here you can see, well, the dominant microbial community members, you can see their relative abundance, and you can also see their relative activity, which is where we use the RNA reads to calculate the relative abundance rather than the DNA reads. Now let's see what else we can do. So we also looked at N converting genes expression. That would be the expression of specific genes using the metatranscriptomics data. So for example, here to the left, you can see the NIR-K expression in, now the units here are transcripts per million. These are actually the number of reads that were mapped to genes that were annotated as NIR-K, normalized to the mag length, and to the entire depth or size of the sample. And you can see here, I think, an interesting thing. You can see here that NIR-K was overexpressed in the aerated phase of the reactor operation, significantly overexpressed. And the second interesting thing that you can see here, that this overexpression is mostly associated with one of the AOB mags. That would be AOB1, which is also the most abundant one among AOB. And then here you can see another example. This is NOR-B expression. Now, this is part of the denitrification pathway. And you can see that as a comparison, there was no significant change in NOR-B expression between the different aeration phases. Here I didn't put a legend, but these different colors represent different mags. And then the reason that we chose NIR-K and NOR-B and also NOS-Z and other genes from the denitrification pathway is because we were specifically interested in the N2O emission by these reactors and who is actually perhaps behind them. Yeah, but I want to focus here on how we can actually get from this metatranscriptomics data, we can get information about the differences in gene expression between different conditions, both at the community and population level. Here, just specifically here, we tested the NIR-K expression and the NOR-B expression. And then another thing that we did, which I think is nice, is that I called it here gene expression patterns. This is actually kind of an overview of all the genes in all the mags, or at least here in these 6 mags, the 2 Anamox mags and the 4 AOB mags. And I'll just go through this briefly. Yeah. So each dot here on this volcano plot is actually a gene. And if the genes are actually behind, above this line here, this horizontal line here, that means that they are overexpressed in the comparison between the aerated and non-aerated phases. And what is interesting, I think, that if, for example, we look at the right side of this figure, so these are the genes from the AOB mags. And you can see this is the percent of all genes of all coding sequences that were identified in these 4 mags. And you can see that all of the mags are, I mean, when we look at the pool of genes, of the AOB genes, so they are kind of equally distributed between the different 4 AOB mags. But when we looked at the genes that they are actually differently expressed, so we see that almost all of them actually belong to mag AOB1, which is again the most dominant one among AOB. And yes, so these are actually genes that will be downregulated in the transition from the aerated to the non-aerated phase. And then when we do the same exercise with the Anamox guild, so we see again, so we had 2 abundant Anamox mags, and we see that if we take all of their genes, so we take the pool of Anamox genes, and so we see that it's equally distributed, very equally distributed between these 2 mags. But when we look at the differently expressed genes, so we see that actually all of them belong to mag Anamox 2, which is actually less abundant than Anamox 1. And we can actually see that these genes are actually, wait a minute, these genes where these genes were downregulated at the aerated phase. Now, of course, if you remember, since we have these mags and we have these coding sequences, predicted coding sequences, so we can also annotate them. And when we annotate these genes, so we find out that when we look, for example, at these overexpressed genes, which again, I remind you, were downregulated at the non-aerated phase, so we see that they mostly belong to respiration, inorganic carbon metabolism, protein synthesis, and folding pathways. And again, when we do the same exercise with the Anamox skills, so we see that these genes that Anammox2, the less abundant Anammox microbe, the genes that it downregulated when the air was turned on were actually they belong to respiration, to either the respiration pathway or had things to do with folding. Yeah. And with that, actually, I will actually end the presentation. So we saw that metagenomics, or the way we define when metagenomics, it includes shotgun sequencing, de novo assembly, and binning. Also, don't forget that you can also recruit or map the reads to reference databases without assembling genomes. And then metagenomics allows us to assemble metagenomes, assemble genomes, again, MAGs, and identify the microbial players and their relative abundance, metabolic capabilities, and interactions. And then with metagenomics and metatranscriptomics combined, we can map the RNA reads to the assembled genomes and identify differences in gene expression at both the community and population level. Of course, if I think that I didn't maybe highlight it or stress this before, but we use the metatranscriptomic data, the RNA reads, with the metagenomics data. I mean, the analysis, our metatranscriptomic analysis was based, of course, on the MAGs that we assembled first by metagenomics analysis, and then, of course, assembling genomes, annotating genes, and then with the RNA reads, we could identify differential gene expression. So yeah, so I hope now, as part and as I said, that was an introductory, kind of a one-on-one to metagenomics and metatranscriptomics, and hopefully you know now a bit more than you knew before, and thanks, guys.
Okay, thank you, Tal. Maybe you can stop sharing your screen at this moment. I'm doing this all, trying to find the formalities. If you can stop sharing.
Yeah, here.
And then
oh wow, that's a very big delay.
What is happening now?
It looks like we hear what is going on downstairs in a delay. Thanks, guys.
Okay, thank you, Tal. Maybe you can stop sharing your screen at this moment. I'm doing this all, trying to find the formalities.
Yeah, here.
And then
oh wow, that's a very big delay.
What is happening now?
It looks like we hear what is going on downstairs and a delay. Thanks, guys.
Okay, thank you, Tal. Maybe you can stop sharing your screen at this moment. I'm doing this all, trying to find the formalities.
Yeah, here.
And then
oh wow, that's a very big delay.
I'm going to stop this because this keeps on going. This is something very strange. Oh, we're gonna stop this. This is bizarre. What?
Thank you, Tal.
I'm going to end and start this webinar again.